and welcome to our lecture on selective and differential media. Here we're going to be looking at 3 different types of media, 2 of which are used in clinical testing and 1 which is used in environmental microbiology. So first of all, when we look at different types of media that are used in laboratories, we oftentimes look at 2 specific types known as selective and differential. There are a few others that we learned about in lecture, but here in the lab we're going to talk mostly about selective and differential. So selective media is media in which different ingredients have been added that allow the growth of one specific type of organism and usually will inhibit the growth of another one. The most common example is allowing the growth of gram-positive organisms but preventing the growth of gram-negatives. This can be done with lots of different types of ingredients and dyes and other types of nutrient deficiencies or additives that again will promote the growth of one type of organism but prevent the growth of another. We oftentimes will want media to be selective because if we are testing a mixed sample, say a patient sputum sample or a wound sample, there's going to be more than one type of microorganism present in that sample. We suspect that they probably have a gram-positive staph infection. We want to see if gram-positive staph are present. So in that case, we might actually use media that would inhibit the growth of any gram-negative bacteria and maybe even some other gram-positives that we know are not necessarily going to be staph. So we can use selectivity in media to be very specific about the microorganisms that we are looking for. Now, differential media is probably the most common, and it is used to differentiate bacteria from each other based on some type of metabolism or growth characteristic. One example that we're going to use here when we look at our selective differential media is that we're going to look at microorganisms that might have the ability to ferment one type of sugar while those other organisms cannot. So let's dig in and look at our examples. It's really helpful to look at the different types of media and what they're used for to really get a good grasp on what it means to be selective or differential. I also want you to keep in mind that all 3 of the media actually that we are going to be looking at are a combination of selective and differential at the same time. So the first media we're going to look at is called the Mannitol Salt Agar, or MSA. This is a diagnostic media. It's used in clinical laboratories around the world. It's very common media. It is both selective and differential, and it is used specifically to look for the presence of pathogenic Staphylococcus aureus. We are looking for the staph aureus that causes the common staph infection. This particular media is selective against bacteria that are referred to as non-halophilic. Just so you know, halophilic bacteria are bacteria that can grow in high salt concentrations, high salt environments. Staph aureus is a common skin bacteria. We find it on our skin. It's part of our normal flora. Our skin is very salty. So Staph aureus that grows on our skin can grow in a high salt environment. There are other organisms that can grow on our skin that are also capable of growing in a high salt environment. So we're going to have to find some way to not only select for our Staph aureus using selective media, but also differentiate that Staph aureus from other halophilic bacteria that are also capable of growing in the same environment. So this media is both selective and differential. It is selective in that it selects against the bacteria that cannot grow in a high salt environment, known as non-halophilic bacteria. If they are capable of growing on the plate, then they obviously are going to be a halophilic microorganism. It is also differential specifically for pathogenic staph. So what we have here is we have bacteria that are capable not only of growing in a high salt environment, so that's the selectivity, but we can also differentiate between different types of staph, between pathogenic Staph aureus and perhaps maybe Staph epidermidis, as shown in this picture here, because of the differential properties between the 2. The media has a differential agent. The differential agent is mannitol. It's a sugar. Staph aureus that is pathogenic has the ability to ferment the sugar mannitol. Other microorganisms that grow on our skin that may be halophilic, like Staph epidermidis here, it has the ability to grow in the high salt concentration, but it cannot ferment the sugar mannitol. So I first select for the staph aureus by inhibiting the growth of non-halophilic bacteria. I then differentiate my staph aureus from other species of Staphylococcus through the fermentation of mannitol. Now, fermentation is an acidic process. So this media actually has 3 ingredients that tell us a couple different things. First is our selective agent. The selective agent in this media is salt. Remember, I am looking for halophilic bacteria only. The differential agent in this media is mannitol. Mannitol is the sugar that Staph aureus is capable of fermenting, but Staph epidermidis, in our example here, is not. Then we have a pH indicator. Because the metabolic property or metabolic process of fermentation results in acidic waste products, we're going to add a pH indicator to our media, which simply just tells us whether or not fermentation has occurred by changing color. In the case of the MSA plate or Mannitol Salt Agar, we use phenol red. Phenol red is yellow in acidic conditions and red or hot pink in alkaline conditions. So if we take a look over here on the right, this plate is acidic. This one is non-acidic, not quite alkaline, and this one is no growth. Okay, so no growth. We can see here, this is Staph aureus. Our plate turns a bright yellow. That tells us that Staph aureus is present. So it has selected due to the halophilic property, and it has been differentiated by fermentation of the sugar mannitol. Mannitol is the only sugar available in the media. So here are some typical MSA results from our laboratory. And in this one, we're looking at mannitol here. We're going to take a guess at what organism number 1, organism number 2, and organism number 3. Take a look at it and think to yourself, which organism is our pathogenic Staph aureus? Well, we know that it has to be able to grow in the media, so it's not really going to be number 1 because number 1 is not growing very well at all, very little. So we're down to 2 or 3. And we know that it has to be able to ferment mannitol. The fermentation of mannitol results in yellow coloration of the media because of the pH indicator. So that tells us that number 3 is the microorganism that we are looking for. If you notice here, number 2, this microorganism is a halophile. It is capable of growing in a high salt concentration, but it cannot use mannitol as a carbon source. So instead, it converts to breaking down proteins. And protein, this is referred to as protein catabolism. And protein catabolism in bacteria results in alkaline conditions. So this microorganism here, number 2, instead of fermenting mannitol, it started breaking down proteins. This results in alkaline conditions, and our pH indicator turns this bright pink. We have the same thing occurring over here. So this is our pathogenic Staph aureus, the one that's turning yellow. This would probably be a gram-negative bacteria because you can see there is no growth. Nothing is growing. And here on the right, this is our non-pathogenic staph. Just right there we go. Non-pathogenic staph. So let's take a quick review. What is the differential agent in the MSA test? Is it salt? Is it mannitol? Is it glucose? Or is it the pH indicator phenol red? In the case of the MSA test, the differential agent is mannitol. We are differentiating pathogenic staph from non-pathogenic staph through the fermentation process through mannitol. What's indicated by the change to yellow in the media? Why does the media turn yellow? Well, it turns yellow because of that differential agent. So here, the answer is C. The mannitol was fermented. And then here we have, this should say question 3. Question 3, we'll fix our typo there. What is the MSA test used to determine? Does it look for group A strep? Does it look for pathogenic Staph aureus? Does it look for halophilic pathogens? Or does it look for fermentation of mannitol? In the case of diagnostics, it is used to detect the presence of pathogenic staph aureus. The next media we're going to look at is called EMB, or Eosin Methylene Blue. This is actually an environmental media. It's used to test for the presence of fecal coliforms in water and soil environments. This particular type of media is a selective media, and it is selective against gram-positive organisms. We are looking specifically for fecal coliforms, and fecal coliforms are gram-negative. These are gastrointestinal bacteria. These are the microorganisms that grow in the colon. That's why they're called coliforms. And in this case, we want to inhibit the growth of gram-positive organisms just to get them out of the way. We're looking for coliforms. Those are all gram-negative, so we're just not even going to deal with gram-positives. It is also a differential media, and it differentiates between fecal coliforms and what are referred to as environmental coliforms. And we'll get to the difference between these two in a few minutes. Now, the ingredients in here, we're going to have a selective agent, a differential agent, and of course, some kind of pH indicator, because we're going to be looking for fermentation. The selective agents, there's actually two of them in this media. The first is called Eosin Y. It's a dye. The second is methylene blue. This is another dye. Both of these dyes together are selective against gram-positive organisms. They break down the cell walls in gram-positives. The differential agent here is lactose. Lactose is fermented by fecal coliforms, but not well fermented by environmental coliforms. So this means that we can detect the difference between a fecal or human type of coliform like E. coli and a what we call an environmental coliform such as Salmonella. And of course, we need a pH indicator of some kind, and these selective agents in this media also act as our pH indicator. Eosin Y and methylene blue in combination, when fermentation occurs, will turn this bright metallic green color that you see here. So let's take a look at some of the differences here. Now, we again are differentiating between fecal coliforms and environmental coliforms. And the reason for this is, let's say we are testing a beach, right? A freshwater lake, it has a beach. People go and they go to this lake and swim in the lake and go on boats and such. And there are, if there is raw sewage contamination, then we're going to see human fecal coliforms present in the water. And that would give us this bright green, metallic green type color that we see here in the E. coli. But ducks, turtles, fish, those types of organisms also live in that environment. And those organisms actually have coliform-like bacteria as part of their normal flora. Salmonella, for example, is normal flora on lizards and also in the gastrointestinal tracts of most waterfowl. So it would be common, or it would be normal, to see Salmonella in a water sample. Now, Salmonella is toxic to us as humans. It causes salmonellosis and food infection, but it is normal in that particular environment. So economically, if we're thinking economically, we would not want to close down a park or a beach because Salmonella was present in the water. That is normal for that environment. So in EMB testing, it's important that we are able to differentiate between sewage contamination, E. coli and fecal coliform presence, and normal coliform bacteria or environmental coliforms such as Salmonella. Now, over here on the left, we have our typical type of results. Here we have this kind of pinkish one. They'll be either a pink color or lavender. This is our environmental coliform. Here on the right, the top right here, this is our E. coli, which is our fecal coliform. And here at the bottom, we don't really see much color change. The bacteria is kind of clear or takes on the color of the media itself. This is a non-coliform, such as Pseudomonas. Pseudomonas is a bacteria that will grow on this plate, but it is not considered a coliform of any kind. It's neither environmental or a fecal. Now, how do we differentiate between these? It has to do with the ratio or rate of fermentation, how much acid is produced during fermentation. So fecal coliforms are referred to as rapid or heavy fermenters. They produce a lot of acids during fermentation, and so that's what gives this bright metallic shiny green. Environmental coliforms are slow fermenters, and they do not produce as much acids, and this is why the color change is less drastic, and we see this kind of pinkish color that we're observing here in this plate. Now, if we take a look, let's look at our center plate here. Would you consider this organism here that I just circled? Take a look at it. Would it be considered a fecal or an environmental coliform? And what about the bacteria growing on this plate on the right? Will we consider this a fecal or environmental coliform? So let's do a quick review. In question number 1, what is the differential agent in the EMB test? Mannitol, well, that was used in the MSA test. Eosin and Methylene Blue are actually pH indicators and selective agents. We are differentiating environmental from fecal coliforms using lactose. Question number 2. What is the result of organism B on the plate in the picture? So organism B is pointing to these colonies here on our plate. Would these be considered fecal coliforms, environmental coliforms, non-coliforms, or gram-positive bacteria? Now, in this case, they look like a light pink to me, so we're going to call these environmental coliforms. What is the EMB testing for? Question number 3. What is the EMB testing for? What are we looking for? Well, you could say we're looking for both fecal coliforms and environmental coliforms, but what we're really doing, if we're testing lake water, we expect to find those environmental coliforms, so we are looking to detect the presence of fecal coliforms. We just do it by differentiating them from the environmentals. The last media that we're going to look at is the MacConkey Agar. MacConkey Agar is used for clinical testing, and it is also used for the detection of coliform bacteria. The difference here, however, is EMB is used in environmental testing and MacConkey Agar is used in diagnostic testing. In diagnostic testing, we are not concerned about the difference between fecal and environmental coliforms because we are looking at patient samples, not environmental samples. So the fact that a coliform is present at all is really just what we are looking for. It is selective against gram-positive organisms, and it does differentiate between coliforms and non-coliforms, but it does not go so far as to differentiate between fecal and environmental. The selective agent in this media is actually bile salts and the presence of a dye, crystal violet. That is missing on my slide here, so I wanted to add it. The differential agent is the same as the EMB test, and that's lactose. Our pH indicator in this case is called neutral red. Neutral red will turn this hot shade of pink, this hot, bright, fuchsia colored pink, in the presence of acids. If our differential agent is lactose, again, we are looking for fermentation. Fermentation results in acidic waste products. Those acidic waste products are detected by the color change to bright pink. So in the MacConkey results, you can see here that we've got bright pink here. This is telling us that coliforms are present. Now, the MacConkey test is most often used in the diagnostics of urinary tract infections. We are looking for the presence of coliform bacteria in urine from the patient sample. So the urine sample would actually be directly inoculated onto the plate. This here is called an isolation streak plate. And the isolation method is used so we can differentiate separate colonies. And in this isolation streak plate, if this were a patient sample, they would be considered positive for a UTI. The presence of coliform bacteria in urine indicates a urinary tract infection. Now, over here on the right is kind of different varying degrees. Here we have a lactose positive fermenter in the middle, and of course a lactose positive fermenter on the right. This is just a heavier fermenter. Here on the left, we have a gram-negative organism, but it is lactose negative, so it is considered a non-fermenter. So let's take a look at our review questions here. Which of the following is not differentiated with MacConkey Agar? Well, we definitely select against gram-positives. We know if it's a non-coliform or not. What we do not differentiate is whether it's an environmental or not. So environmental coliform would be the answer to this question. In question number 2, the selective agent in the MacConkey Agar is bile salts. Crystal violet is not listed here, but it's a combination of bile salts and crystal violet. And in question number 3, what is MacConkey Agar selective against? What is incapable of growing in the presence of MacConkey Agar? And that would be gram-positive organisms. The combination of bile salts and crystal violet actually breaks down the cell wall. So the gram-positive organisms do not grow. So that is the end of the recorded lecture for this. On D2L, you will find a worksheet with a link to a website. You're going to go to that website and answer questions on that worksheet about different types of differential and selective media. Do the best that you can. You shouldn't have to research too much outside of that website. The directions on what you have to do for this worksheet are actually on the worksheet, as well as the link. When you are done with the worksheet, there is an assignment folder in D2L that says in-class activity, differential media. You will upload your completed worksheet into the differential media. It is considered one of our in-class. We were going to do this as groups, but I had to take a personal day and we're not going to be able to do that. So we're just going to do this as an individual assignment to help you really understand the different types of media and how we can manipulate media to be selective and or differential in order to tell us different things about bacteria. So do the best that you can on those worksheets, turn those in, and they will be due by our next lab period. Thanks for your patience. Good luck on the assignment. If you have any problems, be sure to shoot me a text. Talk to you soon.